We will now discuss vaccine immunology. In this section of the lecture, we will cover vaccine immunology. So the whole idea behind vaccination is to cause somebody or to induce an immune response, a memory immune response towards a certain pathogen. Kind of the holy grail in vaccine medicine is to cause an individual to develop both T and B cell memories towards a pathogen without experiencing any of the negative symptoms and effects of the disease. So vaccines can be either prophylactic, protects against possible future exposures. These are the typical vaccines that you tend to think of. These are the ones that you give the babies, right? The other type of vaccine is called a therapeutic vaccines, and these are all experimental for the time being. There are a couple of vaccines that can be given post-exposure, like the rabies vaccine, for example, but by and large, a therapeutic vaccine would be something like an experimental cancer vaccine that are just starting to be developed and starting to be used to try to combat certain types of cancers and autoimmune diseases, but there are no real effective therapeutic vaccines that are on the market just yet. So let's talk about active versus passive immunity. A little bit of vocabulary. Active immunity means that an individual produces their own immune response. This is something that, say you get sick, get a rhinovirus, you produce an immune response to that rhinovirus, and at the end of the response you're going to have memory cells against that rhinovirus. That's active immune processes. So this is slow to develop, but it's long-lasting because of these memory cells. And this is exactly what you want with a vaccine. You want to give somebody something that produces these long-lasting memory cells. However, with vaccines, you don't want to get them sick, so that's kind of the catch for that one. Passive immunity, on the other hand, is using preformed antibodies that are transferred from one individual to another. So think something like IVIG, or think something like mother-to-fetus IgG placental transfer or breast milk IgA transfer. Rapid protection, you get protection right away, but it's short-lasting. Once those antibodies die, there's nothing left to protect that individual, and there's no memory there. So instead of having passive immunity where you would have to go get antibodies every time you got sick, we tend to try to make vaccines that promote active immunity. So here's a question for you. A 3-year-old boy receives an antiviral vaccine that has been demonstrated to produce lifelong immunity. 10 years after the vaccine, the boy becomes ill with the disease produced by the virus against which he was vaccinated. Which of the following is the best explanation for this occurrence? Is it A, the vaccine failed to produce an immunogenic response? Is it B, the vaccine's antiviral effects have been reduced due to loss of memory cells? C, the virus has mutated at the spot of the epitope used in the vaccine? D, the virus has developed sophisticated anti-immune machinery? Or E, the virus has obtained an envelope? The answer to this question is C, the virus has mutated at the spot of the epitope used in the vaccine. So in attacking these types of best explanation type of questions, the way to go is process of elimination. So let's go through all of these answer choices. How about A, the vaccine failed to produce an immunogenic response? Would you think that would be correct if it had been 10 years since the vaccine and the boy hadn't been sick? Probably not. How about B, the vaccine's antiviral effects have been reduced due to loss of memory cells? Again, this is a vaccine that it says in the question has been known and demonstrated to produce lifelong immunity, so it wouldn't make sense that this kid had loss of memory cells unless he had some other type of disease. For D, the virus has developed sophisticated anti-immune machinery? Nah, viruses aren't that smart. They don't do that kind of thing. And E, the virus has obtained an envelope. Well, now you're talking about some viral evolutionary mechanism that is not likely to have occurred in this 13-year-old kid. So that one can be eliminated too. C makes a lot of sense. If the virus mutated at the spot of the epitope, which viruses are known to do, then if you only gave this child one epitope in the vaccine, then that epitope would no longer be functional. The immune response against that epitope would no longer be protective. So C makes the most sense. So let's go through the different types of vaccines that are available, starting with whole organism vaccines. The first vaccine is a live attenuated microorganism vaccine, and this is the vaccine that actually most closely resembles an actual infection. It causes a limited infection in the host, sufficient enough to mount an immune response, but not sufficient enough to cause the disease. Because it mimics an infection so well, it provides the most effective vaccination mechanism. You get cell-mediated immunity, you get humoral immunity, you get memory cell development. It's exactly what you want with a vaccine. Some of the dangers are herd immunity or reversion. Because these are live attenuated microorganisms, they do have the ability to revert back to their pathogenic state. So if that were to be the case, for example, with the Sabin polio vaccine, instead of injecting somebody with attenuated polio, you'd be injecting them with live polio. Is that what you want? Absolutely not. So there are some downsides to this, even though they give you the best overall immune response. They're not as safe as the other vaccines. Now, killed or inactivated microorganisms is kind of the next step down from live attenuated microorganisms. Instead of leaving them alive and just weakened, we kill them off. We use heat, formaldehyde, or irradiation to try to kill off these microorganisms. Once they're killed, we know they can't infect. We know they can't revert. We know they can't cause any systemic effects, any symptomologic effects in the person. However, these will vary greatly in efficacy. For one, you don't get a CTL response if it's a virus. Why? Because that virus is non-infectious. It doesn't get into the cells anymore, and you won't get endogenous presentation. So you won't get memory cytotoxic T cells. Other things you have to think about. If you're killing these microbes with heat or formaldehyde or irradiation, well, you're denaturing their proteins, right? If you denature the proteins, it's going to be harder or less likely that the B cells will recognize them. Because remember, they like antigen in its native conformation. So therefore, you may not get as good humoral immunity either. Examples of killed microorganism vaccines include the Salk polio vaccine, rabies vaccine, flu vaccine, pertussis vaccine. Another type of vaccine is a subunit or conjugate vaccine. The subunits or toxoid vaccines refer to, for example, toxins that have been modified by formaldehyde or heat. So take something like tetanus or diphtheria. You isolate the toxin and then you modify it so that it's no longer toxic. At this point, we call it a toxoid. You can then inject this toxoid into somebody and it will get an immune response, evoke an immune response. Again, like the killed vaccines, the killed microorganisms vaccines, they're not quite as effective as it would be if you injected somebody with the regular toxin. However, you can't inject somebody with tetanus toxin. That's a bad idea. So the toxoid subunits are fairly effective enough that they're very useful in our society. Conjugate vaccines, we take microbial polysaccharide antigens, for example, and couple them to protein carriers to elicit T helper cell responses and thus achieve high affinity antibodies. So in this case, think of something like the Hib vaccine, where they use a capsular polysaccharide. They couple that to a protein carrier that will then be broken down by an APC, presented to a T helper cell, and we get nice cell-mediated immunity. We can get nice humoral immunity with high affinity secondary type antibodies as well. Another class of vaccine is called the recombinant vaccines. So let's look at recombinant antigen vaccines. Here we have, or we take, microbial antigens known to invoke protective immunity, and we clone them into an expression vector. We isolate the gene for the protein that's known to be immunogenic. We then take that gene, we clone it into an expression vector such as E. coli or yeast, and we have those bacteria or those yeast mass produce this protein. Then we take that protein, we isolate it, purify it, and we vaccinate people with it. An example with this is the hepatitis B vaccine. Another way that we can do this, kind of along the same vein, is using recombinant vector vaccines. In this case, we use live viral vectors to induce a cell-mediated immunity. We take those genes, like we did in the recombinant antigen vaccine, we take the gene that's known to produce an immunogenic protein. But in this case, instead of just making a lot of protein, we insert the gene into a virus, a live virus, and then we take that live virus and we inject it into somebody. Now, this live virus has been previously treated so that it won't be infectious and won't cause symptoms, but it will be able to replicate that gene that you put into it. It will then produce the protein intracellularly because it infects the cells. It just doesn't cause damage. That protein will be expressed on MHC class 1, and then we can get a nice cell-mediated immunity. So an example of this would be the vaccinia virus. Synthetic peptide vaccines are another recombinant vaccine. In this case, instead of isolating the gene and having some type of biologic entity make that protein, like a bacteria or a yeast, we just isolate the gene, we get the amino acid sequence, and then we produce it by machines. It tends to be a little bit more pure, it tends to be a little bit more controllable, but it's far more expensive than, say, having E. coli or yeast make the protein for you. So here's a question. The influenza vaccine initiates which immune response? Is it A, humoral, B, complement mediated, C, cell mediated, or D, innate? The correct answer here is A, humoral. The influenza vaccine, remember, is a killed microorganism vaccine. So therefore, it's non-infectious. It won't get into cells, and therefore you won't get that endogenous presentation. You won't get a cell-mediated response as much as you would a humoral response. A humoral B cell will pick up that antigen. Even if it's denatured, it can pick it up. You will get B cells that will pick it up and can process it and promote a humoral response. So A is the best answer there. This concludes our discussion on vaccines. In the next section, we will discuss hypersensitivity.